This is a little review of our exam 1 material, a little bit of chapter 1 and then chapter 12 on the nervous system. So we first started off talking about the difference between anatomy and physiology. So remember, anatomy is the study of structure and form. I like to think of anatomy as what it is, where physiology is the study of function. I like to think about how things are working. One of the things that I had to keep in mind this semester is that form equals function. When you see bones with different shapes, it's because they have a certain purpose. So on the exam, I will give you examples and you will have to identify whether it's anatomy or physiology. So remember what versus how, and you can actually do some practice on your own with that as well. So kind of think of some different questions on your own. Another thing that we talked about were the characteristics of living things. All living things undergo metabolic processes, and under metabolic processes, we talked about catabolic, breaking things down, and anabolic, building things up. Generative processes, this is the big umbrella that includes reproduction, growth, also healing as well. Responsive, living things are going to respond to the environment. Living things are also going to respond internally. And that kind of brings us to that homeostasis, that balanced internal environment. To have those control processes, we need to be able to maintain that homeostasis. Finally, living things have structure and organization. Now, a lot of things have structure and organization, so that alone does not make something living. But what it really comes down to is being the cell. This is what we call the cell doctrine, that all living things are made of cells and all cells come from other cells. So that really is the crux of something being living. Now, one of the things that you want to practice with is if, let's say that you take an object, maybe a stuffed animal, and you have to describe why it's not living. Or like a frog, something that's living, describe why is it living, using these different characteristics of living things. So this is another thing that you can practice with. Talking about that responsiveness and control, we talked about homeostasis, which is a big theme in anatomy and physiology. So we talked about positive feedback versus negative feedback. Positive feedback is that self-amplifying cycle. So continuing into that greater change in one direction. So this is just going to continue. Classic example is childbirth. Contractions build and build and build until the baby is born, and then we go back to where we were in the beginning. Blood clotting is another example. So one clotting factor goes to the next, until we actually have that clot form and we have that internal Band-Aid. So they often call this self-amplifying because it's this greater change in one direction. This is a little different from negative feedback. There are probably far more examples of negative feedback in the human body, regulating blood glucose levels, body temperature, blood pressure, heart rate. This is what I kind of compare to that teeter-totter, so that up and down. So we have our set point. So for example, our blood pressure, 120/80. Now we know that we're not always 120/80 every second of the day, and our body has to make adjustments. So the first thing that happens is that there's a stimulus, something changes. So either our blood pressure goes up, goes down. We have to have receptors or sensors to sense that change, send it to the control center, and the control center is usually someplace in the brain. And then we need to figure out what to do. And then the control center sends out the orders to the effectors. So for example, in our example of blood pressure, perhaps those effectors are the heart. And so the heart could either lower the heart rate, raise the heart rate, depending on whether or not we needed to lower blood pressure or raise it. Another effector could be blood vessels. If we open up those blood vessels, blood pressure will decrease. If we constrict them, blood pressure will increase. So there are several examples that you could actually use. Don't forget with our negative feedback, we have to have these three parts: receptor, or also called the sensor, control center, which is also called the integrating center, and then the effector. Then we got into the nervous system. So the nervous system, we first started off by talking about the setup of the nervous system. So central nervous system, this is kind of like the big computer of the body, the brain and spinal cord. Everything outside of that is what we call the peripheral nervous system, the PNS. Now, one of the things that this picture really isn't showing us is that communication between the central nervous system and the peripheral nervous system. So I gave you a little acronym: SAME, sensory, afferent, motor, efferent. So that sensory information is going to be picked up by the peripheral nervous system, sent to the central nervous system by the afferent system. So I think of that information as arriving. Arrive begins with A, afferent also begins with A. Then the central nervous system figures out what to do and then sends that message through the motor efferent system. So I like to think of that efferent system as that information exiting the central nervous system. Now, within the peripheral nervous system, we have the sensory division and motor division. The visceral sensory division is going to pick up that information internally. So think about those organ systems: cardiopulmonary, digestive, urinary. This is from the viscera, right? So if you have a gut reaction to something, you're having a visceral reaction. So internally. Somatic sensory division, we're talking about muscles, skin, bone, joints. Then we have our motor division. Now in A&P1, we're mostly in the somatic motor division. This is sending voluntary information to our skeletal muscles. So I think of somatic skeletal muscles. This is going to be voluntary control so that if we need to pick up something, our muscles are going to allow us to do that. Our somatic motor division is going to allow us to do that. What is going to be involuntary is our visceral motor division. And remember, our visceral motor division has an alternate name. This is called our autonomic nervous system. It literally means self-governing, and things are going to happen automatically. This is also when we talked about the autonomic nervous system as having two divisions. Our sympathetic division, also called our fight or flight, versus our parasympathetic division known as rest and digest. Now, oftentimes I kind of think of the parasympathetic division as being kind of that calming voice. So if, let's say that you went outside, thought you saw a bear, think about physiologically what's happening. Heart rate increases, breathing increases. What's not important is digesting what we had for lunch. So our sympathetic division is going to get us ready for this crisis situation. So do we either stand our ground or flee? Now imagine that we see, maybe it's not a bear, maybe it's a large dog. Now we're starting to feel more relaxed. Breathing rate, heart rate comes down. This is our parasympathetic division kicking in, that voice of reason, and that's our rest and digest. It will also direct those maintenance systems to do their job as well. So we first started off talking about the setup of the nervous system. Then we started talking about some of the players in the nervous system. So we looked at the structure of a neuron. This picture that you're seeing here, this is a multipolar neuron. So you can see there's a soma, many dendrites, and then we have the axon, and then we have an ending with those axon terminals. This is the most common neuron. We see this a lot in the central nervous system. So this picture right here, I'm going to guarantee you that you are going to see on the exam. So you want to make sure that you can recognize these parts. I'm not going to make you label it. However, there will be multiple choice questions where it will ask you, based on a number that's pointing to the certain neuron part, what that part is. So be familiar with these pictures. We also talked about the supportive cells. So these neural glial cells, or also called glial cells, they are going to outnumber those neurons by about 10 to 1. So you also want to know the functions of each of these. Remember, there's 2 in the PNS. They both are S words. There's 4 in the CNS. This was also on your study guide as well, so make sure that you are acquainted with those. The last thing in the part 1 of chapter 12, it is a large chapter, that's why we divide it up into 2 parts. We talked about the regeneration of a nerve fiber. It's possible, if all of the criteria are met, only in the peripheral nervous system. And that's because of that Schwann cell and that neurolemma. The other thing is that the soma needs to be intact because that has all of the parts and the machinery to make all of those repair parts. The brake has to be very close. If it's too far apart, those ends aren't going to meet up. And then they also have to be on the same level. Now, this is not perfect. Sometimes it can take months, even years, and that neuron probably isn't going to have the same response that it once did. So that's a little bit on the regeneration of a nerve fiber. Then in part 2, we started talking about the communication, because remember, the nervous system is all about communication. So here you see 2 neurons. So we have our presynaptic neuron and our postsynaptic neuron, and they are communicating. So I like to call the presynaptic neuron the sending cell, the postsynaptic neuron the receiving cell. So one of the things that I mentioned in this chapter is that communication is going to go from electrical to chemical, electrical to chemical. When we're talking about electrical conduction, it has to do with the ions. When we're talking about chemical transmission, we're talking about neurotransmitters. So this got us thinking about that communication. We already talked a little bit about in the axon terminals, those neurotransmitters are going to be present. But in this chapter, we got a little more involved in that electrical transfer. So this has to do with a membrane electrical potential. And we first started talking about the resting membrane potential. There's more sodium ions outside the cell, more potassium ions inside the cell. So potassium tends to have a bigger influence just because those potassium ion channels are kind of leaky. So remember that sodium-potassium pump that takes a lot of energy to run. In fact, about 70% of the energy our nervous system needs comes from running that pump. We constantly need to restore that ion balance. So we talked about the resting membrane potential of the neuron being minus 70 millivolt. When we get into the muscular system, you'll see that the muscles, their resting membrane potential is a little different. So this is that membrane potential. And as those ions move, then this is when it's going to change and we can actually start to send that electrical signal. Now, one of the things that we talked about, how do we open up these ion channels? We talked about ligand-gated channels. This is when a neurotransmitter would bind. That's building up that local potential. That's going to open up those ion channels. We also talked about voltage-gated channels. So that's when that change in electrical potential in front of that ion channel changes and it snaps open. We mostly see that with those action potentials as it's traveling down the axon. Now, this picture right here, this is where we have that chemical signal. So in order for that presynaptic neuron to release the neurotransmitter, no matter what that neurotransmitter is, this is the process that is going to be the same. So when that action potential hits that axon terminal, it opens up calcium ion channels. Calcium flows in through cell signaling. Calcium tells those vesicles to release the neurotransmitter. Neurotransmitter flows across the synaptic cleft. Then we have our postsynaptic neuron on the other side with receptors, and the neurotransmitter binds to those receptors. Remember, this is where those ligand-gated channels are. So no matter what we're talking about, excitatory or inhibitory, this has to happen in order for neurotransmitter to be released. So one of the things that you should know is that whole sequence of events of release of neurotransmitter and then generating that local potential to that action potential. So you should know those events. All right, here's where we're building up our local potential. So now we've got those ligand-gated channels, neurotransmitter binds, and ions are going to start to move. Now, one of the things that we talked about, if it's excitatory, that's going to be more depolarizing, bumps us up closer to threshold. If it's inhibitory, that's going to put us in a valley where we're going to be farther away from that action potential. So if, let's say, that stimulus is strong enough to generate a local potential, it's built all the way up to that axon hillock, so that neck region where that soma is going into the axon. If it's strong enough, now we're going to open up those voltage-gated channels, and this is where that action potential travels down that axon. Now, that change in electrical potential is because we've got sodium ions moving in, potassium ions moving out. So they're constantly trading positions, and that's what's going to send that impulse down the axon. Now, one of the things that you should be able to even draw out and recount is the action potential graph. So this picture is showing us what's happening in the membrane, also what's happening with the action potential graph. So you need to be able to label this action potential graph and discuss what's happening. So resting membrane potential, our ion channels are closed. When we begin depolarization, sodium ions come in, and that's when that peak starts to increase. So that line is increasing. Then we're going to open up potassium ion channels. Potassium leaves. Now we're becoming more negative. So now we're going back down the other side of that hill. That's what we call repolarization, because those potassium ion channels are slow and leaky to close. Some of the potassium kind of sneaks out, and then we're a little more negative than resting membrane potential. This is what we call hyperpolarization. So be able to recount the action potential graph and know those different phases and also what's happening in each of those phases. Now, one of the things that we talked about with action potentials is that there needs to be a little pause before we can generate another action potential. So this graph is showing us the difference between absolute refractory period and relative refractory period. During absolute refractory period, absolutely no way can we generate another action potential. Does not matter how strong that stimulus is, because everybody is doing what they're supposed to be doing. We have to wait a little bit until we're on the other side in relative refractory period. Now, we can generate another action potential at this point, but it needs to be especially strong. If you see where that yellow bar is, we're in that hyperpolarization. We're in that valley. So the reason that that stimulus needs to be especially strong is that we need a bigger jump up to threshold. So it's possible at that point, it just needs to be very strong. The other thing that we talked about are the different types of synapses. There's over 100 neurotransmitters, but we really only talked about 3 synapses and 3 types. So this picture is just a little reminder that we have that communication. presynaptic neuron communicating with that postsynaptic neuron. Remember, electrical, chemical, electrical, chemical. So when we're at the synapses, this is when we're talking about the chemical, so the different types of neurotransmitters. So this picture right here, we are seeing an excitatory cholinergic synapse. This is using acetylcholine, where acetylcholine is excitatory. That means it's more likely to generate an action potential. So as we talked about, the release of neurotransmitter has to start with that action potential, zapping that synaptic knob, that axon terminal, opens up calcium ion channels, calcium goes in, cell signaling happens, neurotransmitters released. In this case, it's acetylcholine. It's going to bind to receptors on that postsynaptic neuron. These are associated with sodium ion channels. So remember, those are ligand-gated channels. When sodium comes in, we become more positive, and now we're depolarizing. So depolarizing is going to bump us up closer to threshold. So that's why in this case, it's excitatory. The inhibitory neurotransmitter we talked about was GABA. So again, in order to release GABA from that presynaptic terminal, zap that axon terminal with that action potential, calcium ion channels open, calcium goes in, release that neurotransmitter GABA. Now, again, we've got our ligand-gated channels. However, this ion channel is not associated with sodium. It's associated with chloride. This is a negative ion, an anion. So when chloride goes in, now we're becoming more negative. So now we're becoming in that hyperpolarized state. It's a bigger jump up to threshold, so less likely to generate. That's why it's inhibitory. Our third synapse, this is an adrenergic synapse. This is not associated with an ion channel, but the secondary messenger system. So we talked about that neurotransmitter norepinephrine binding. The receptor is associated with a G protein, and this kicks off a whole cascade of reaction. With this secondary messenger system, it can be amplified because we've got all of these messengers running around the cell. More to come on this because we're actually going to be seeing this in the endocrine system. So be able to discuss each of these synapses and whether one is excitatory, whether one is inhibitory. So if you like to draw pictures, draw this out. If you can look at these pictures and you can talk about what's happening, you're going to be in good shape. As I always say, if you can explain it, then you know it. If you want to write it out, you can. If you want to draw the picture and write it out. So be familiar with that. All right, so the whole thing about this communication is that we need to be able to start communication and stop communication. So we need to clear the signals so that we can have a little bit of flexibility. So we talked about how we can clear that neurotransmitter. It's not going to be hanging out in that synaptic cleft forever. So it's just going to diffuse out to those neuroglial cells that are going to absorb it. It can actually be taken back up into that synaptic knob by that way of endocytosis. That's the opposite transport form than exocytosis, the way that it's released. And we can also have enzymes degrade that neurotransmitter. So for example, acetylcholinesterase is in that synaptic cleft and it can degrade, break down that acetylcholine so it's not hanging out there forever. So remember, it's all about control. It's all about flexibility. That's what the neurotransmitters get for us. Now, there are places in the body where we have electrical synapses so that we have ions flowing between cells. This is going to be quick. The problem is that we lose a little bit of that flexibility. That's okay in some places. So in some places in the body, we have gap junctions. So for example, in the heart, we don't want our heart to stop beating. So to get that signal to go from one cell to the other, we have these gap junctions between the cell. So it allows for quick transmission, but we're losing that flexibility. And that's okay in some parts of the body. Now, one of the things that we talked about is that there is a lot going on with that postsynaptic neuron. It's actually getting a lot of information. By using those chemical neurotransmitters, that helps with that flexibility. It also helps with decision making. Yes, it's a little slower than those electrical synapses, but it allows us to have a little control. So, one of the pictures that I showed you is this neuron that's receiving lots of information from other neurons. Some of those signals are going to be go, excitatory, some are going to be stop, where they're inhibitory. So it's kind of like the loudest noise is actually going to get that result. So there needs to be this process of integrating this information and figuring out whether or not we should have an excitatory response or an inhibitory response. Now, some of the ways that this communication can happen is that we could actually have one neuron that is just firing fast and furious, and that's in that top picture, the temporal summation. So there's one neuron that's just firing fast and furious, and that postsynaptic neuron is getting the message that something needs to be done. It can also come in other forms from other neurons. Even though you notice on the bottom picture with spatial summation, those lines are spaced farther apart, there's kind of strength in numbers. Because that postsynaptic neuron has 3 presynaptic neurons talking to it, it's getting the message loud and clear that something needs to happen. So we could have one neuron that is sending that stimulus like crazy, or we could have strength in numbers where we have several neurons sending that communication. So if that is excitatory and excitatory postsynaptic potential, that's going to bump us up closer to threshold. If those signals coming in are inhibitory, that's going to be inhibitory postsynaptic potential. It's going to bump us down closer to hyperpolarization. So this is all part of that flexibility and that decision making. Are the signals coming in more excitatory, or are they inhibitory? And that's what helps to give these chemical synapses a little bit of flexibility. So this is a little review of everything that we covered in chapter 1 and chapter 12. As I mentioned, if you can explain it, then you know it. Practice, practice, practice. I like to write things out. Other people like to draw things out, say things. Explain things to anybody that will listen to you, your dog, your 4-year-old, anybody, because if you can explain it, then you know it.